Welcome to Grow with Biology. Today's topic is some terms related to DNA. First is denaturation and then hybridization and TM, that is temperature melting or melting temperature of DNA. Hyperchromicity. Now we start with a solution of DNA. If that solution of DNA is exposed to 100 degrees Celsius or high pH, then the DNA start denaturing. Now what does this denaturing means? Denaturing means both the strands will start separating. If the strands have been separated and then that solution is slowly cooled, then again both the strands will again form a double helix. Means when we started a double stranded DNA, there was hydrogen bond between both the strand which got broken up due to increase in temperature or increase in pH. As both the strands were separated, it is termed as denaturation. And if it is slowly cooled, be specified that it is slowly cooled. If it is normally cooled, then it cannot give rise to its original form of DNA. If it is slowly cooled, then it will give rise to the double stranded or the double helix. Why? Because if it is slowly cooled, then the hydrogen pairing will be very much appropriate or exactly the same which was present before, because there will be no segregations of the DNA. Now the next term is hybridization. When we said that increase in temperature will separate both the strand, at that same point if we interchange the DNA strands, then there are chances that some new strands can be incorporated to the original strand. That will form a hybrid structure, that is known as hybridization. Now how this denaturation can be measured? In 1950, it was measured by taking the absorbance. Now, how this absorbance function? Absorbance at UV range, the DNA absorbs maximally at 260 nanometer. Now, what happened when there is denaturation of DNA? Then at this specific nanometer, that is 260 nanometer, there is less absorption, approximately 40% less absorption. I have mentioned temperature increase. Temperature increase, that is increase in temperature, will denature the DNA, and when that denatured DNA has been taken the absorbance at 260 nanometer, we will find that there is less UV absorption, and this less UV absorption is known as hyperchromicity, and this hyperchromicity is due to base stacking. Means if there is denatured DNA, the absorbance value or the optical density values will be high. Now if there is a graph plotted between temperature and the optimal density or absorbance, we find that to some extent, to a specific temperature, there is maintenance of the double stranded DNA, but after sometimes there is transition of double stranded DNA to single stranded DNA, and the center point or the midpoint is known as TM, that is the melting temperature, where exactly the double stranded DNA convert to single stranded DNA. Now on what factors this melting temperature depends? Melting temperature basically depends on two factors. First is the GC content. Remember, between guanine and cytosine there is 3 hydrogen bond. So the first factor is GC content and second factor is ionic strength. Now if there is more GC content, then the temperature, that is the melting temperature TM, will be high. If there is high salt concentration, then also it will be high. So only two factors on which the melting temperature depends is GC ratio and high ionic strength or ionic strength of the solution. Now specifically, the backbone of DNA consists of phosphate, and each phosphate is negatively charged. When we talk about two strands of DNA, if they have denatured, each strand will have a negative charge due to the phosphate group, and both the strands being negative will repel each other and will favor the conversion of double stranded to a single stranded DNA, that is the denatured DNA. Hope this will clear your doubts about melting temperature and the denaturations. Thank you.